Welcome, everybody, to our February WASP mini conference, Western Apicultural Society of North America, featuring Dr. Dale Hill, who is an animal nutritionist. And I am very pleased and honored to be your host for the evening. I'm Melanie Kirby, and I'm a board member. I serve as the New Mexico director and also on the board as the vice president for WASP. We'd also really like to give a big shout out and thank you to our sponsor for this evening, which helps to cover our Zoom licensing for the Saskatraz hybrid project in Saskatraz, Canada. For those who may not be familiar, these are some of the faces behind the scenes. And we need to get Chris's picture up here too as well, who's our IT tech guru. But our executive board of directors, Etienne Tardif is our president, and he's actually away on business, hence me being the host tonight, or the MC, but he will be with us next month. And yeah, thank you so much to our mini conference committee as well, which is Etienne, myself, and Dr. Karen, and the many others who are a part of the WASP family who help to make suggestions on speakers. If you or someone else you know is interested in doing a presentation or would like to recommend somebody, please reach out to us. We'd love to hear from you. So tonight's program is going to be divided into two parts. We've got Honeybee Nutrition Basics. Dr. Hill's going to cover that for about an hour. Then we'll take a little 5-minute bio break. And then we'll resume with part two, looking at nutrition and feeding management for a little over half an hour, and then we'll do some Q&A at the end. So feel free to put your questions in the chat if you'd like, or even the Q&A section, and we'll be monitoring those for the duration of the program. And with that, I will stop share. And Dr. Hill, you're very welcome to take the floor, and we are so excited to have you. Let me actually read a bio for him. Dr. Hill is an expert in the field of animal nutrition with 38 years of nutrition formulation experience, and is the primary developer of Dadant's AP23 pollen substitute. Dr. Hill is also the author of the updated nutrition chapter in the latest edition of the Hive and the Honeybee, and he also teaches the nutrition section of the Montana Master Beekeepers course, and he has made numerous presentations on honeybee nutrition. So we're very excited to have Dr. Hill with us here today. So please welcome Dr. Hill.
Thank you. Okay, so these are some of the things I want to discuss tonight, and that's basic nutrition. And a lot of what my comments will be is applies not only to honeybees, but mammals as well. And so we're going to talk about water, we're going to talk about protein, actually really about amino acids. We're going to talk about carbohydrates, both starches and sugars, lipids, which are the fats, oils, and waxes, the vitamins and minerals, and what does all this mean and how does it really apply to bees and beekeeping. And then in the second part, we're going to talk about supplemental feeding and management. And a lot of these questions, especially on the supplemental feeding and management, are coming from my own experience and the questions that have come up in the Master Beekeeper course, the nutrition section of that over the last several years that I've been teaching that. And I also should say that right now I don't have any bees. I moved to, I see there's a few people on from Illinois. I moved to South Dakota about 2 years ago. And, you know, they say that beekeepers, there's two kinds of beekeepers, those with bad backs and those who are going to get bad backs. Well, my back is still okay, but I've blown out both shoulders. And I had my second rotator cuff surgery 5 weeks ago today. And I was originally supposed to give this presentation in January, and I talked Jerry into letting me do it in February instead. I've got about 1 more week to go in the sling, and then I start physical therapy. So I'm kind of out of actual beekeeping. I'm more of a mentor now than I was before. So that's kind of where I'm coming from. Now, a lot of the things, or most of the things I'm going to talk about tonight, have been published in American Bee Journal back in 2017, and it's a series of 6 papers starting in May. And I didn't get the September one in by the deadline. So it goes May through November, but you can use that as your reference, as one of your references. I think that's would be beneficial for everybody to go back and read those. And along with that, the Hive and the Honeybee. When I did that, redid that chapter on honeybee nutrition, oh, geez, 7, 8 years ago now. There was in the previous edition, in the 8th edition, or the I think it was 2005 edition or something like that. There were significant errors in when they made the printing, and I got all those corrected, and I pretty much doubled the size of the chapter on nutrition. But some of the others that I want to bring your attention to would be Somerville in publication in 2000, Nutrition and Supplemental Feeding. He's in Australia. Broge Snyder and Karlsheim, they're in Munich, Austria, in 2010, Nutrition and Health. De Groot back in 53 from the Netherlands was one of the first ones to come out with the amino acid requirements at that time. And then Haydak over the years out of the University of Minnesota published several papers. And then Randy Oliver, I know he's on tonight, and he does a pretty good job of keeping everybody informed on the nutrition side. And he's always a great reference. So I recommend his website as well. So we'll start with water, and that is absolutely the most important nutrient for all species. And with most species, if you don't have water, they are dead within 3 to 4 days. And if you go through the literature, you're going to find that 5 to 6 gallons per hive per year is the general estimate of water needs. And I think that if you get down into Melanie's area, into the southern US and down into Central America, that may be a little bit of an underestimate. I think they may actually need more than that. Part of that is used for nutrition purposes and maintaining the bodies of the animals. And part of that, especially during the summer months, is used for evaporation and temperature control within the hive. So my recommendation is always to provide clean, fresh water if the hives are not close to a water source. This helps with reducing the flying distances, helps with the bee efficiency, if you will. And by close to the water source, we're generally talking within a quarter of a mile or half a mile, so they don't have long distances to fly. The challenge is that the bees don't read the books. They've been around for over 1 million years. We haven't been around quite that long, but a lot of the bees seem to prefer the less clean water sources. They prefer the ponds, sometimes the stagnant water. It really makes you wonder if there is something to the taste of that water, or a bacterial population that they're trying to get, or whether it's just a behavioral issue that they are landing on moss along the edges and there's a reduced chance of drowning. I don't think we really know, but it's pretty obvious that the bees seem to prefer the less clean water sources. Okay, let's go to amino or proteins. And first of all, the amino acids are the building blocks of protein. And the way to think of this is as a log chain made up of many, many links. And the links are different colors, kind of at a random assortment. And when you digest that protein, there are specific enzymes that will only cut that chain at specific color combinations. So, and then the way the body utilizes it, you can kind of think of that as the same way. Now, I don't use protein level when I do product formulas. The only reason I use protein as part of my calculations is for product labeling and make sure that they meet the legal requirements on labels. I really don't care what the protein level is. I care what the amino acid levels are, because that's what is the important part of the whole picture. And just like mammals, the bees need 10 essential amino acids that must be provided in the diet, plus 10, we'll call those non-essential amino acids, that the bees or the bacteria in the gut can make in adequate amounts to meet the metabolic needs, or they are always in excess of what's required in common pollens and artificial diets. Now, when ingested, the proteins are broken down by broken down to sometimes individual amino acids by these digestive enzymes. Sometimes it's 2 or 3 amino acids in a chain. And depending upon the molecular size of those, they either have to be individual amino acids or 2 or 3 or 4 amino acids in a chain to be small enough to be absorbed. There's a maximum for, at least on the ruminant side, of about 600 Daltons for molecular weight to be absorbed. Anything larger than that typically is not absorbed. Once the amino acids are absorbed, they're transported to specific tissues where the amino acids are reassembled into body proteins and enzymes in the appropriate sequences for the body's needs. Now, realizing that this is not a very efficient process, but this is Mother Nature's way of maintaining species differences between proteins. And this is where the DNA, RNA for each protein comes into play. So that's again, that's not a very efficient way to do it, but that's how Mother Nature maintains those species differences. And so the concept of limiting amino acids or limiting nutrients is that if we think of this barrel and each stave as a requirement, and each stave goes up to the point where it's whatever is provided in the diet, or I should say whatever is required by the body and whatever is supplied in the diet. So if the staves are the requirement for the body and the water, let's say the water is the amount that's in the food sources, it's pretty clear to see that whatever nutrient is in lowest supply relative to the needs of the bee is what is going to be able to, the bee is going to be limited on their maximum productivity because of the concentration of that one nutrient. So whatever nutrient is in the lowest supply relative to need, that is the one that's going to limit the productivity. That is the brood production, the royal jelly production, the foraging ability, and so forth for these bees. So in worker bees and drones, the hemolymph, or the blood, if you will, the hemolymph for the is the equivalent of blood in the bee. That amino acid level increases and peaks at about 5 days of age based on the pollen and the bee bread consumption. And then it decreases as the bees age. That is, as the bees get older and go from nurse bee to guard bee to forager, their protein stores in the hemolymph will decrease. The queen, on the other hand, will reach her maximum amino acid in the hemolymph at about 10 days of age, and that usually coincides with the onset of egg laying, and it remains about 2.5 times higher than the worker bees. And this higher amino acid levels are really needed for egg laying activity. And along that line, it never ceases to amaze me when we talk about the ability of the queen to lay eggs, because she can, at maximum production level, maximum egg laying production level, she can lay a close to half of her body weight in eggs per day. And that's like a 1,500-pound dairy cow producing 250, 300 pounds of milk a day. That's just unheard of. It's just mind-boggling from an animal nutrition standpoint that the bees can do this, the queen can do this. So we can't overstress the importance of protein or amino acids and the other components as we go through here when we talk about the efficiency of egg laying production by the queen. So an amino acid deficiency, what we know, will decrease productivity and increases susceptibility to diseases and especially to toxic chemicals. And if you go through the literature, you'll find that about 5 days of non-foraging can lead to cannibalism of the larvae in the worker bees. So that means 5 days of non-foraging. We're talking about 5 days of rain, 5 days of weather where the bees can't fly, 5 days of extreme pollen and perhaps nectar dearth. So when this happens, when you get those 5 days or a week of very poor weather conditions, you need to check your frames and see if you have day-old eggs or 2-day-old eggs. I've come across my frames where I've had no eggs in the middle of the summer. And so the first question you have to ask is, do you have a queen? And if the queen is present, why isn't she laying? Is she injured? Is there inadequate pollen? Is there cannibalism because of weather, because of inadequate pollen being brought in? Are there other factors? You have to watch for this, especially during periods of pollen dearth. It may be in extreme conditions, you may need to supplement even midsummer with protein for the bees. Now, the diversity of pollen sources is very important for bee foraging. There is no single pollen source that meets all the amino acid needs of the bee. And we'll come back to this a little bit later. But it's the same story for lipids. And lipids, we're talking about fats. That's the equivalent of what we talk about on animal and human nutrition. And so should we supplement during pollination? And the answer is probably yes. If you're on a single pollen source for more than about 2 to 3 weeks, you probably should consider supplemental with a pollen substitute. And again, the pollens are the major source of protein, or the amino acids, while the nectar is a major source of sugars, or the carbohydrates, but it is a very minor source in most cases for the amino acids. Now, the nurse bees will feed the queen, so they also need to have higher protein, because of when the queen's laying eggs, they need somewhere in that 18 to 20% relative protein. The foragers need a high carbohydrate, that is a high sugar, high honey food intake, and relatively low protein. And the recommendation there is about 4 to 5% protein in the total diet. And some references even suggest that for foragers, all you really need is the sugar and the honey. And as long as everything else is going in good condition, you know, the overall environment, your apiary is bringing in pollen, that's probably fine if you only have to supplement with the sugar water for a nectar source. We know that winter bees are more efficient in pollen protein digestion than summer bees, and this strongly suggests that the protein level during broodless periods should be lower than during egg laying periods by the queen. So we know the pollens will vary from about 4% up to about 60% crude protein based on nitrogen analysis. But the problem is that crude protein value really does not give us any indication of the digestibility or the biological utilization of the proteins, that is the amino acids. So why do we use protein for this? Well, the reality is that you can run protein for $4 or $5 per sample, and amino acid analysis typically will be between $200 and $300 per sample. So that's why in most cases, we go back and look at protein levels rather than amino acid levels. Okay, reduce pollen availability. If they're not bringing in pollen, we know that's going to reduce the brood production. We know that there's a lower need for pollen in late fall as the bees transition to winter bees and the egg laying is decreased or has stopped. Late fall winter bee supplementation depends on the pollen stores within the hive for winter survival and the condition of the bees. And yeah, when we talk about condition of the bees, what we're really talking about is the protein or amino acid stores in the fat bodies of the bees. And this is kind of a judgment call on the beekeeper's part. How good has the weather been? How much pollen has been brought in? What are the stores within the hive, within the colony that when you're going into winter? I've had years that I put protein supplements on my bees in mid-August because things weren't looking real good as far as pollen stores and honey stores in the colony. So I fed them heavy in October or in August and early September, and I figured they had enough, so I pulled it off. And by mid-October, they were just about out of groceries again. So you really have to be watching as a beekeeper the food stores within the hive. So in some cases, I think the fall supplemental feeding of the higher protein is justified, especially some of the commercial guys where they have to have the bees in good shape for pollination in the almonds in California on late January, early February is when they ship them out there, and they are paid on the strength of the colony. So I think in some cases those guys may want to consider pushing some extra protein to those bees in August and September. So in general, the bees will harvest pollen whenever it's available and from whatever plants are in bloom. So this is and I'll come back to this later, but that brings up the point of we need to make sure that there is a good variety of plants in bloom throughout the foraging season. Now, bees will generally not visit grains, that is the corn, oats, wheat, barley, and the other wind pollinator plants that are from the grass family, except during severe pollen shortages. And with that said, I will also say that they may visit some of the hybrid corns. I've heard of a few variations or varieties that the bees will forage on, but for the most part, they will not touch corn, but they will forage on sweet corn. So kind of keep that in mind. Okay, adult foragers will have a shorter lifespan on higher protein. There is no negative effect that we know of if the foragers consume only the sugar and the honey. The risk of death increases with excess proteins as the worker bees age, that is, as they convert to foragers. And again, this is not the case with the queen. She has to have that high protein requirement as long as she's laying eggs. And the non-laying queens will probably be fed less total food by the workers over the winter. So they will still, I think, feed her I know they will still feed her the royal jelly, but it's the total intake, her caloric needs and protein needs are going to be decreased. And I think that's controlled by just total less food intake or less food offering by the workers to the queen when she's not laying. And the drones, once they reach sexual maturity and start flying, they have a minimal protein requirement and they survive just on the honey that they are fed. Now, I threw this in here. This is from Wikipedia, and these are pollen grains. And if we look at, we'll show this one here. If you look at these center indentations, that's a fairly soft spot where the digestive enzymes can get in and break through to get to the inside of that pollen shell. And what you will find in feces is pretty much empty pollen shells. They don't have the gut bacteria is not there to break down these pollen shells. So you will find the pollen shells, empty pollen shells in the feces of the bees. And also I have a warped sense of humor, but if you look at these, especially when you look at some of this or some of these, the spiny edges on here, that is the plant's way of attaching to things and being able to move around and pollinate other plants. But it's also great for attaching to nasal passages and causing some allergic responses. Okay, let's move to carbohydrates. There is no true requirement for carbohydrate, but there is a true metabolic need for glucose, fructose, and precursors that can be metabolized via the Krebs cycle and converted to honey. And the excess amino acids can be used as an energy source, but it is metabolically less efficient and is much higher protein or much higher metabolic cost to the beekeeper, much higher metabolic cost to the bee and out-of-pocket cost to the beekeeper. And in some cases, protein toxicity may be a concern. This excess amino acids and the fact that it's poorly utilized is the basis for a lot of the human high protein weight reduction diets like the South Beach and some of those. So starches and sugars, the difference is in the chemical bonds, and that creates some structural differences. The starches are not well utilized by honeybees. There are no specific enzymes to break down these chemical bonds. And that's the same story with mammals. Most mammals, well, I shouldn't say it that way. They have, depending upon the species, they have amylose, which is the enzyme that digests starches, breaks the chemical bonds. And it depends on the age and physiological stage as to how well those starches are digested, how much amylose or other enzymes are metabolically available to do that. So the sugars are metabolized for energy for flight and activity, and these, especially the sugars, are mixed with enzymes in the honey stomach. They're regurgitated and then dehydrated to make honey. Now, sucrose is 50% glucose, 50% fructose, and this is a very good carbohydrate for bees. This is the primary sugar that is found in nectars. And from what I can find, the bees have to have preformed glucose and fructose. It does not seem like they can convert some of the other sugars into glucose and fructose to make honey. So they have to have the glucose and fructose from the nectar to be able to make honey. They can also do that with high fructose corn syrup, which is a little bit different ratio of fructose and glucose, but it's still a good carbohydrate source for bees. I'll come back to high fructose corn syrup in a minute. Now, honey will average about 38% fructose, about 30% glucose. And all of these are easily metabolized by the honeybee. One of the challenges is that many bee researchers will use sugar to describe the, when they're doing their research, they'll just say they use sugar. Well, in many countries, there's several different sugars that are used interchangeably. And if you don't clearly define in the scientific journals which specific sugars that you're using, it's really hard to try and figure out the effect of some of this research. And this is much more common in some of the foreign journals, especially coming out of Africa and the Middle East, where they have much more limited resources to work with as far as laboratories. And they maybe sometimes they don't have the facilities or the funding to clearly define or get the types of sugars that they really need them. So that's sometimes a problem when you go through the literature. It does not always specify what type of sugar that they're using. Okay, some of the sugars that we must avoid is dextrose, ribose, ribinoxylose, mannose, galactose. Lactose is a milk sugar, and then the oligosaccharides. And these are compound sugars, complex sugars that the raffinose and stachyose are typically found in some of the seeds, the seed proteins, that is, such as soybeans. And these are the ones that can cause diarrhea, especially these. And even in the animal world where I work, we limit the amount of soybean meal that's used in diets because of these two sugars. You will start, if you get too much in there, it will, in livestock production, you will actually back off growth rates. The reason these are a problem is that the bees lack the enzymes to digest these compounds, and some of these may be actually toxic to the bees. Some will result just in diarrhea because it's the bacteria in the gut that can digest these. It's not the animal or the bee itself. So use only sucrose or commercial, that is food grade high fructose corn syrup when feeding your bees. Now, I'm going to back up to this one. With high fructose corn syrup, well, I think I've got a slide. I'll come back to that one. Some nectar and honey is used for food sources for yeast and bacteria when they're fermenting the pollens in the bee bread for later use. That's their food storage. And in this fermentation process, it's if you get a good fermentation, it's much like making yogurt or sauerkraut or the alfalfa corn silages that we feed the livestock. If you get a normal fermentation, acetic acid is the primary preservative. And if you get the wrong bacteria or something goes wrong with that fermentation, you will often get alcohol production. So while that may create some healthy bee or some happy bees, it's not healthy for the bees. And I'm gonna leave it at that. It's the other comment I was wanting to say here is, if you are looking at your food stores and your pollen, and if you see a dark, dull brick red pollen, I always remove that because that is usually an indication of a poor fermentation. And I just use my hive tool and scrape that out and let the bees redo the wax and put down new wax and go from there, making new bee bread. But it's only the dark dull red bee breads that are potentially toxic. So the sucrose of nectar will vary from about 5% up to about 75%. And obviously the bees are efficient workers. They prefer the high sucrose nectars, and they will seldom visit the nectar sources that are less than 5% sucrose content. And you can find some of these sucrose content from the different nectars, the different flower sources, and some of the books by Eva Crane and some of the other books that are available. Now, nurse bees have very limited ability to digest raw starches from grains. So my recommendation is to limit the use of grains and supplements for feeding bees. I know there's some out there that are primarily grain-based, and I very much disagree with that approach. I don't think that that's good use of resources and good for the bees either. Now, the foragers may be able to digest more starches, but still that needs to be very limited. On the mammal side, if we cook the starches, they are much more digestible. We break a lot of those chemical bonds, and that gelatinized starches are much more digestible. I don't know whether, I've never seen any cooked starches in any of the bee supplements, the labeling that I've looked at. One of the problems is that the starch overload will usually result in diarrhea. Again, the bees are going to have very limited ability from an enzyme standpoint to digest starches, and it's going to be more of the bacteria in the gut that digest these. And that if you get too much starch in there, all of a sudden you get a proliferation of the bacteria. You will get a lot of acid production in the gut, and that's usually the cause of diarrhea. So there's a small amount of starches that are sometimes used as a flow agent in sucrose. just to keep it from caking, and that's usually less than 0.5%. So it's usually not a problem. Okay, bees prefer high sucrose nectars, and these come from members of the rose family, the legumes, the mint family, the figwort family, the mustard family, and the aster family. And these are the 6 major nectar producers for honeybees. And these are it's plants from these families that you want when you do your pollination plots. Honeydew is also available in certain cases, and this is an excretion product from insects. And chemically, it's fairly close to most nectars. I want to spend just a little bit of time on cell membranes. And the cell membranes of the queen are highly monounsaturated and have a low polyunsaturated fatty acid content. And this is the same story with newly emerged bees. And when we talk about highly monounsaturated fats, think about a beefsteak or a pork chop when it's coming out of the refrigerator and how hard that fat is, and it's very structural. That is a monounsaturated fat. That's saturated monounsaturated fats. So it's going to give that queen good cell structure, good solid cell structure that is fairly rigid. Now, within the first week in the newly emerged worker bees, that polyunsaturated fats will increase and the monounsaturated fats will decrease. And the polyunsaturated fats, think more like vegetable oils. There's really no form to them. But this as it changes from the monounsaturated to the polyunsaturated, this gives the cell walls a little bit different 3-dimensional configuration. And it also increases the susceptibility of that cell wall to lipid oxidation, or in other words, rancidity is what I'm looking for. So it's the same chemical reaction where they're talking rancidity or oxidation. And this is likely a big factor in the differences in lifespan between worker bees and the queen. And any disruption of that cell membrane will have a negative impact on health and longevity. We know that lipid oxidation is likely a contributing factor to aging and cell death in all species, and it's been implicated in some of the cancers as well. Just as kind of a sidelight, banana oil is very similar to the guard pheromone. So the moral of the story is don't eat bananas before you go work your bees. Okay, pollens appear to provide adequate lipids under normal conditions, that is normal weather conditions. And sometimes it's kind of hard to define normal weather conditions, the way things are going. And maximum lipid digestion occurs about day 8 and then tends to decrease with age. There's very little fat storage in insects as compared to mammals, and the fats and amino acids are stored in the abdominal fat bodies along with amino acids, enzymes, vitamins, and minerals. And that's also the reason why, if you've heard Sam Ramsey's presentations on the varroa mite going after the fat bodies, I think that's a big reason that the varroa mites are doing that, because they want these same nutrients for their reproduction. Okay, nurse bees can synthesize mandibular acids, and this the mandible is in the human is the jawbone. So just under their jaw, they have glands that synthesize specific amino acids or specific acids. These are fatty acids actually from acetate, and they produce this. The one that's the most important is called 10-hydroxy-2-decanoic acid. And this particular fatty acid is not found in any plant or pollen sources that I've been able to find in the literature. And the reason that is so important is that it is an essential fatty acid in royal jelly. And it may be as much as between 2 to 5% of the royal jelly by weight. And this fatty acid is essential in ovary development and ovary function. And nurse bees will lose this ability to make this compound as they transition to foragers. But the real question is, do they lose that ability as they transition, or do they transition because they lose that ability? And I think it's the latter from what I can find in the literature. As they lose that, as the age and they lose that ability to make that 10-hydroxy-2-decanoic acid, that is the metabolic trigger for them to transition to foragers. And there's very low efficiency if they have to, if the foragers have to transition back to nurse bees. What I've found in the literature suggests that that's somewhere around 10, maybe 20% efficiency. So if you're doing queen bees or grafting, make sure that you have plenty of nurse bees. And the way to do that is to pull those nurse bees out during midday when most of the foragers are out of the hive. Okay, the queen bee cannot synthesize this 10-hydroxy-2-decanoic acid in her mandibular gland, so she must be fed the royal jelly, and that's why you always see so many nurse bee attendants with the queen. So again, when grafting eggs for queens or when splitting colonies, make sure that you have adequate nurse bees, or the developing queen may not have fully developed ovaries, or she may not be able to lay enough eggs for a good brood. So that brings up the question, and it's one that I always bring up to the class at the Master Beekeeper course, is queen failure may not always be the fault of the queen. There are several management factors that can go into that as well. So there's also a second gland secretion that goes into the royal jelly, and that it comes from the hypopharyngeal gland. And the pharyngia is the tongue. So this is right below the tongue. So both of these are in the head of the worker bee. And the hypopharyngeal glands are clear and they're mostly protein. Again, the mandibular glands are white and they're mostly lipids. So the worker bees, or I should say the worker larvae, are fed the worker jelly, and that's made up to 20 to 40% of the lipid material for the first 2, maybe 3 days while they're in the larval stage. And then they are switched to only the protein material mixed with honey. And the worker bee is, the larvae are somewhat limit fed. On the other side of that, I'll go back to this, because on the other side of that is that the queen is fed that royal jelly, roughly a 50-50 mixture for her entire lifetime. Okay, there's the approximate composition of the royal jelly. It does have antibacterial and antibiotic effects. Some of these compounds are not storage stable, so we must use fresh royal jelly when queen rearing and queen grafting. The royal jelly is a high protein, high lipid, high vitamin content. It's high sugar content, and this likely determines the intake of royal jelly. Glucose is the primary sugar needed for queen development, and there's no satisfactory royal jelly substitute that has yet been developed. And I think Randy would agree with me that if we look at that, we need to have a royal jelly or even a protein supplement or pollen substitute that will support more than 2 generations of bees. So the queen larvae, again, are fed equal amounts of the lipid and the protein material throughout larval development. The lipid components are responsible for the queen development, that is the pheromones and the reproductive hormones. She needs a source of cholesterol, or the bees need a source of cholesterol, and that is, in plants, it is a 24-methylene cholesterol, and that is very low levels. And if they don't have enough of that, they can't make enough of the the decanoic acid compound. And this is unlimited feeding to the nurse bee, or by the nurse bee to the queens. The primary protein in royal jelly is vitellogenin, and this is a glycolipoprotein that's similar to poultry egg protein. The worker bees will deposit this protein in the glands of the head and abdomen during larval and pupal stages, and it is metabolized very slowly after emergence. And the current thinking is that the worker bees transition to foragers as they become deficient in this protein. And we also, there's some information in the literature that would suggest that this also has a role in juvenile hormones. So we know the pollens are rich in water-soluble vitamins, that is the B vitamins, and they're poor in the fat-soluble vitamins, that is the A, D, E, and K. So it's safe to assume that the vitamins play the same basic biochemical role in bees as they do in other species, with the one major exception of vitamin D, and that is that vertebrates have an internal skeleton that's based on calcium, phosphorus, magnesium, and calcium absorption and utilization is primarily regulated by vitamin D. Now, I'm going to hedge just a little bit because in the last 4 or 5 years, they have found multiple other roles of vitamin D, and perhaps there is a somewhat of a role in bees for vitamin D. So insects have an internal skeleton. Back up. Insects have an external skeleton, which is called the exoskeleton, that is chitin-based. And chitin is a sugar-based compound. It's fairly similar to cellulose. It's the supporting structure of plants. But vitamin D has no known role in this process. Vitamin E likely has the same biochemical function as a cellular antioxidant. It helps reduce oxidation of fatty acids, and it is likely a determining factor in the aging process of all species. And vitamin E as a cellular antioxidant also plays a role in immunity, and that is the ability to resist diseases in mammals. It's very likely it has that same role in bees. A and D for humans and animals are chemically stabilized by esterification. And there's nothing in the literature that I have found that would indicate that the bees have these specific enzymes to break this ester bond and to use this more chemical stable form. Therefore, in the supplements that I put together for bees, I use the less stable plant forms that are more water soluble or lipid soluble, but they are, the A is basically a beta carotene, and the E is a tocopherol, or mixed tocopherols, which are used as antioxidants in a lot of foods. So they're still, in the concentrations that we use, they're still pretty stable. Vitamin C, or ascorbic acid, is a cellular antioxidant and also regenerates vitamin E. And no known use for E in bees except maybe to regenerate the vitamin E that I've been able to find in the literature. Now, the question is how much of each of the B vitamins is really needed to support the bee life functions, especially the reproductive role and the survival of the brood. And it's very limited scientific information out there. It's very difficult research to do. It's very expensive. And to do this type of research, you have to use purified diets. So you know the exact chemical composition, and trying to get that type of a diet that the bee will eat is extremely challenging. I have not, in the work that I've done with development of the AP23, I have found that they will not consume purified diets. As far as the mineral side, very little is known. Again, it's a very expensive, time-consuming, challenging area to work in. Most of the published requirements are based on collected pollens and not on the bee body composition or the physiological response. And that, I think, is a major problem. So I don't think we have a very good handle on where we need to be for the vitamins and minerals. it is reasonable to assume that there's the same biological role in insects as mammals, except for the skeleton. And livestock minerals that are designed for high calcium, high phosphorus, high sodium are not ideal for bees. Okay, so make sure, as far as feeding the queen larvae, make sure that you have frames of honey and pollen close to queen cells, and for brood rearing and nukes or new colonies, so that the food sources are readily available to the nurse bees. And the recommended feeding source for sucrose, or the recommended feeding source for queen larvae or queen production, is sucrose rather than high fructose corn syrup, because there is more wax production from sucrose than there is from high fructose corn syrup. And the literature that I've been able to come across, there is greater bee body weights when you feed the sucrose. But this really depends on economics and labor costs. I think for most of us smaller guys, the sucrose is probably the better way to go. For the commercial guys, probably when they can automate the feeding process, anything that they can do to cut labor costs, that's where the high fructose corn syrup comes in. They can afford a little bit less production just based on their labor cost. Now, much of the scientific information out there regarding bee nutrition is based on pollens collected in pollen traps and analyzed, and that's based on the incorrect premises of you are what you eat. And although that's somewhat popular in some areas of research, or some areas of nutrition, that really is not a very good scientific approach. Some of that pollen is not digested or utilized, so it's better to say you are what you don't excrete, and this is the much better approach, but it still does not provide the best evidence for nutrient needs. The nutrient needs really need to be based on body composition. And when I developed the AP23, I actually took over a pound of bees out of several different colonies, froze those, freeze dried them, ground them up, sent them to a lab for amino acid analysis. And that's what I based a lot of the AP23 formula on, is that type of body composition. You also have to consider specific metabolic enzyme systems that require specific nutrients. And this is very hard to do on bees. It's much easier to do in the animal kingdom than it is with, when you're working with poultry and swine and cattle. It's much easier to do than working with a bee. You got a much bigger animal to work with. It's much easier to get blood samples. So this approach, again, is not without faults, but it's a much better method of determining the nutrient needs. So that brings up the question is, are there differences between summer bees and winter bees? And my best guess is there probably is, but I have not found what I would consider good information in the literature to really know what these differences are. As far as measuring the biological response, its evaluation of pollens and supplements for the biological potency should include longevity and the amount of brood reared on test diets, the hemoglobin protein or amino acids, the phytogenin levels, changes in weights and nitrogen content of the bees fed to test diets, and the colony weights. And as far as supplemental feeding, some of the ingredients, I'm just going to touch on this because I'm going to spend the next half hour on the second presentation going into quite a bit more of this. But there are big differences in soy. And if you read the labels on the protein supplements for bees, make sure that they are using soy protein concentrate or soy protein isolate instead of the soy flour. Remember what I said on the raffinose and the stachyose. Those are removed when you go to the soy protein concentrate or the isolate. And it's the same story if you see pea protein products. Make sure it's the concentrate or the isolate. And when you see yeast on the labels, I will say that there are big differences in yeast sources and suppliers and how that yeast is, the yeast sources and how it's processed. The bees will readily consume some yeast sources, others they will not touch. As far as direct-fed microbials, they may be useful under some stress conditions. Make sure that you don't feed direct-fed microbials and antibiotics at the same time, because the antibiotics will typically wipe out the, especially the terramycin will wipe out a lot of the bacteria or the direct-fed microbials. But the bigger question is the commercially available direct-fed microbials are intended for livestock applications. And the livestock eat a very different diet. So the substrates for that bacteria are very different in livestock, poultry and livestock, than it is for the honeybee. So I know there's some work being done around the country on the microbials that are found in the bee gut, and those are not the same as the commercially available direct-fed microbials. The other comment there is that rate of passage, that is from consumption in the mouth through expulsion through the anus in the bee, is about averages about 2.5 hours. And the regeneration time of most of the direct-fed microbials that's on the market are, the lactobacillus, the bifido, are in the range of 8 to 10 hours. So my question is there enough time, residual time in the gut for those bacteria to have any real effect? And so my recommendation is, if you want to use direct-fed microbials, make sure that you are on a record-keeping system where you can do a good economic evaluation of direct-fed microbials as to whether it benefits your bees or not. Because I guess at this point, I'm not totally sold on the fact that they may be, in some cases, they may be beneficial, but I think in most cases, they probably are not. Again, on the grains, the raw starches are probably poorly digested by the nurse bees and foragers, but they are digested by the gut microbes, at least in mammals. And we're just now starting to look at the microbial genome in the bee gut. I don't know how well those bacteria are going to digest starches. If there are starches in the supplements, if you're feeding that while the bees can make cleansing flights, it may not be a problem, but in winter feeding, you may want to watch and make sure that you're not getting a lot of extra diarrhea. You're going to always have to watch for brood patterns and brood amounts. They may be able to use those raw starches for fermentation of pollens to be bred. I don't know. I've not found that in the literature to be clearly defined. Cooked starches may be okay, and I put big questions marks behind that. It's improved starch digestion in poultry and mammals, but I don't know whether it is in bees. Okay, animal proteins. Some of the examples here would be eggs, albumin, animal protein or animal plasma, casein. These are all excellent amino acid sources. I would be careful with the casein because that, even though the lactose is supposed to be removed from that, there may still be some residual lactose in there. These are all going to be relatively expensive protein sources, but they will have very good digestibility. Blood meal is not as good. The bees will forage on dead carcass animals. And if you go back to the ancient Egyptians, they thought that the bees actually originated from dead animal carcasses. It's probably an acceptable protein source from the bee standpoint, but the bees are not the ones buying the products. So I don't think it's going to have a very good acceptance with the beekeepers. You do need a cholesterol source, and dried eggs work very well for that. You don't need a whole lot in there, so you don't have to add a whole lot of cost to that. Plant sterols are, I say, very expensive. I'm going to say prohibitively expensive, and probably not used as efficiently as the the cholesterol from the egg. Okay, the high fructose corn syrup. The one big precaution with high fructose corn syrup is a product called HMF, and that is hydroxymethylfurfural. Easy for me to say, right? But hydroxymethylfurfural is a natural degradation product that starts to form at about 105 degrees Fahrenheit. And during long-term, it forms naturally during long-term storage, but even faster if you've got prolonged exposure to the sun. And heat above 105 will increase the rate of hydroxymethylfurfural production. One of the things that happens is that if you look at fresh high fructose corn syrup, it is nearly a clear solution. As the hydroxymethylfurfural starts to form, it will start taking on a brownish tinge to it. So if you are a commercial producer, make sure that you know what your hydroxymethylfurfural content is. And if I remember correctly, I think you want to keep it less than about 20 to 25 parts per million hydroxymethylfurfural. If it gets much higher than that, I would not use it. Invert sugars, we've got the same concern about the hydroxymethylfurfural. Sucrose, use either cane or beet sugar. There's really, as far as the bees are concerned, there really is no difference. The difference from our standpoint is that the cane sugar is probably going to be non-GMO and the beet sugar will probably be GMO. Now, that's, in my book, that's a personal preference. I don't think it makes a bit of difference to the bees. So the point is that you need to know what you are buying. If you are buying surplus or damaged food ingredients, there's a lot of that out there. Just make sure you know what you're buying and know what you're getting, and know what the challenges are with some of those. So for most of us, it's going to be best to stay with the sucrose and the high fructose corn syrup. Again, don't use livestock minerals. It's the wrong mineral levels for bees. It may actually attract deer, bears, raccoons, and other wildlife. And I've probably had more raccoon problems in my 15 years of beekeeping than any other wildlife critters. If used, the livestock mineral supplements, if used, use only the vitamin minerals that are formulated for bees and make sure it comes from a reputable supplier. The minerals should be either in a sulfate, in a chloride, in a proteinate, amino acid chelate forms. Those are the preferred forms. If there's oxides there, they are poorly utilized. And then again, if the label says vitamin A acetate or vitamin E acetate, I'm not sure that the vitamins will be available to the bee. So we don't have as many details as we need and as we have for basic livestock species. We do need additional government research dollars worldwide for nutrition and disease research. The point being is that we must have a balanced diet. There's several pollen sources and or supplemental feeding. Make sure they have access to water. And the more you get into beekeeping, the more you know, and the more questions you ask, and the more questions that you ask, the more you realize that we don't have all the answers. But that's part of research, because research answers one small part of the puzzle at a time. And I think Randy will agree with that. So I think that with that, we'll stop and take a biology break. I did want to put this picture up. This picture was taken at the Alamo in San Antonio several years ago. And it's not so much that the fact that this is not a honeybee in there, but the fact that there's pollen sources all over the place, and you just need to look for them sometimes.
Okay, so a few of the questions, this one coming in, the first one I see up here from Lorena is saying, I have been told Drivert sugar is a preferred late winter supplement for low food stores. How is it different from plain sucrose? And if grains are generally not good for bees due to the diarrhea or dysentery, why do bees go for the corn dust left from feeding birds? Good question.
Well, let's first address it's invert sugar, and invert sugar is basically sucrose that has been treated with acid, with hydrochloric acid, and it's used mainly in food processing. It can be used in place of sucrose if you can get it. It's probably going to be a little harder to work with, depending upon how many colonies you have and what facilities you have. Is it worth any additional cost? I don't think so, because the bees will actually cleave that glucose fructose bond during their digestion process anyway. So it can be used. My only concern would be the hydrochloric acid content, because I'm not sure how much acid is used in the invertase or inversion process. And then the second question was on the corn droppings, or the corn milling, the leftover feed. Yeah, they'll go to that. But I've also been out in my shop working with woodworking, and the bees will take sawdust back to the hive as well. So I don't, as long as it is something that is I think pretty close to the appropriate micron size that they're looking for food. If they're hungry, they will probably take it back and see if they can utilize it. Other than that, I don't have a good answer. But I do agree that they will do that. But they again, they will also take sawdust or other things back to the hive as well.
Yeah, I think an acceptable answer is just, bees do the darndest things, right? Some
Again,
things.
they don't read the books, but on the other hand, they've been around for several million years.
Right. And we're still learning what they're doing. Okay, another question we have is, so are the new supplements like the probiotics and the super DFM, which you mentioned, digestible? Are they needed by honeybees?
Well, again, I think if they may be useful in cases of extreme stress, that's where we use them on the livestock side, and only during certain life cycle phases where we want to increase feed intake. But again, if you want to use those, I would strongly suggest that you have a good record keeping in place, so you can do your own evaluation of these and see if you are getting an economic return. The probiotics, you may be able to get an economic return. Well, I should say the prebiotics, you may be able to get the prebiotics are the food, basically the food for the bacteria in the gut. And the probiotics are the actual bacteria. But you have to have a good record keeping system in place. And that means weighing hives on a regular basis, or doing your hive inspections, keeping very good records to really see if the cost that you're putting out there for those products, if you're really getting an economic return. Because I'm not convinced in most cases that you are, because it's not the same bacteria as in
Right.
Dale, if I might
Let's
jump
let Jerry.
in.
Jerry's got his hand up. Go ahead,
No,
Jerry.
I don't know how I got my hand up after that. I think Zoom helped me and put it there. Anyway, I just want to jump in on this. We've had a couple different groups in our U of M online master classes for their projects look at some of these super DFM and other products. Generally, what they found, doing what Dale said, keeping good records, weighing highs and so on, weak stress colonies that were showing some type of problem, at times seem to get a boost from some of these products. On the other hand, if they were healthy, normal looking colonies, they either saw no difference, or in some cases it almost looked like it had a negative effect, slight, but so it doesn't seem to be a major problem feeding them. But the only time that any of those projects showed any real significant effect was, again, colonies that were not growing well, or that were leggy, or had some type of problem, or were under some type of nutritional stress. So I think the analogy that Dale made with the cattle and so on applies also, from what I can see from those. And then they're not, the end all, but they've consistently come up with that same type of pattern. So, and again, you really have to be taking detail. The other thing we learned from those projects, you really have to have a well-organized, well-replicated, and rigorous system of weighing or so on, just to be able to evaluate whether you see a difference or not. So the biggest problem with some of these is it's, the old, throw it on and the bees look better, so you assume that it works and so on. It may or may not have.
Right.
Again, it goes back to how much stress they're under, and if you can provide that to them specifically during that time of stress, there may be an economic benefit, but otherwise, I really doubt that there is.
Okay, well, and we of course have more questions than we have time at the moment, but I want to ask just a couple here, and then hopefully, it'll come up in your next presentation, or even at the end, there'll be an opportunity to answer some of these. But Skip mentions here, dirty water research by Rachel Benowen seems to indicate that the honeybees do prefer quote unquote dirty water for some mineral nutrients and value. Do you have any comments on that? Are you familiar with that?
If he could send me that reference, I would greatly appreciate it.
Yeah, great. I only recently heard about her, so I'm not overly familiar either, but it's something I want to look up too.
Yeah, because it makes sense, but I just don't have any documentation on it.
Right. Let's see here. How long does the 24 methylene cholesterol last in stored pollen?
Probably not more than a year.
Okay.
And I would say that if you're going to collect pollen and store it for later use, it needs to be frozen in airtight packages and should be used the next season. It 2 years in a freezer, you're going to lose probably 10 to 20% of the effectiveness, and by 3 years in a freezer, you're probably going to lose about 75–80% of the effectiveness.
Okay. Yeah, there's a lot of really good questions. I want to ask all of them, but I want to still hear the next presentation. Okay, one more, and then we'll try and come back to these questions as well. So let's see here. This one's interesting. What ratio of egg should they use? Because too much eggs cause swarming. I don't know if that's more of a comment, or I think that you're talking about proteins.
Right. In a supplement, probably somewhere between 1 to 2%. You don't need a lot of egg in there. You're mainly using it as a cholesterol source, not as a protein source. And you're not trying to overwhelm them with cholesterol, so it doesn't take a whole lot.
Right, and this is sort of a related question that I'll just tack onto it, and then we'll start your next part 2. But this question is regarding the egg protein. Do you use the yolk or the white? Do you use it cooked? This person mentions that they know raw egg whites tie up other proteins in the gut of vertebrates. Same with raw soy.
Well, it actually, the raw egg white will tie up biotin, one of the vitamins. And what is commonly used, what I use on the livestock side and what I used in the beef supplement, is what they call a dried egg product. And it's basically dried whole egg. So it has both the yolk and the white. So the cholesterol is, and the protein is, or most of the protein is going to be, I should say most of the lipid components are going to be in the yolk, and the egg white is basically albumin. So it's very highly digestible, and it is a cooked product.
Great. All right. Well, let's go ahead and move on to part 2, and then we can come back to these questions at the end.
I think some of the questions may be answered as we go through the second part of this.
Yeah, and you also got a really nice comment that they really liked your presentation, a wonderful presentation too.
Well,
so.
I'm kind of on the fringes of the way most speakers would present, but because I put a lot of information in the slides rather than just talk about it. And the reason I'm doing that is I know that there's at least one from New Zealand that I saw online. And when you go overseas, or even in the US in some places, the audio and the video don't always coincide very well. So if you missed one side or the other of it, that's why I got a lot of that information on the slide. So hopefully, if you go back and look at this later, you can get the gist of the conversation, not just tidbits, but you can get most of it. Okay, so the
Great.
second part of this is feeding management. And a lot of this comes from comments, again, that I've had from students going through the Master Beekeeper course in Montana. And so I'm going to talk a little bit about hive inspections and what I want you to be looking for, seasonal feeding suggestions. We're going to talk a little bit about FLUR, or the infrared photography, and the winter bee clusters. And then I want to spend a little bit of time on plants for bees. So on the hive inspections, and I know this is going to be redundant for a lot of people, but again, when you're looking for that queen, if you can't find the queen, do you find eggs that are 1 to 2 days old? And if you don't find those eggs, kind of keep in mind what I talked about the cannibalism during pollen dearth. So when you look at that brood pattern, is it pretty solid with just a few empty cells, or is it a spotty pattern with lots of empty cells? What are the pollen and bee bread honey stores in there? Is it a normal deposition pattern within the frames and on the foundation? And then always, is there any indication of disease? So we'll keep on going here. Are the foragers bringing in pollen? The easiest way to do this is stand out in front of your hives, off to the side, I should say, don't stand right in front. But what are they bringing in for? You can see that on their hind legs, and it's really pretty interesting to see their landing pattern sometime. And actually, it's easiest if you video that and then come back and look at it later, because it can be rather amusing to watch as well. Are the bees consuming the sugar syrup? Now, one of the things when you're feeding sugar syrup, in my experience, is that early on in the spring, when you're starting new packages, or even nukes, or feeding during midsummer during a pollen and nectar dearth, if you're feeding syrup and they're consuming that syrup and as far as syrup consumption, I've seen them take in very little, or I've seen them take in 2 quarts a day. But my point is, if they are consuming syrup and within 24 hours, it goes from good syrup consumption down to nearly zero, or essentially zero syrup consumption, you better be looking for a queen, because my experience is when that happens, you have lost a queen. So that's kind of one thing to keep in mind while you're feeding syrup. So what's the general population of the colony and the brood pattern? Has the weather impacted the pollen and nectar sources? Pollen dearth, cannibalism, keep those factors in mind. And based on the overall condition of the colony and the weather patterns that you're seeing, do you need to have supplemental feeding? I took this in central Iowa a few years ago, and if you want pollen dearth, here it is. You got a cornfield on the right side, you've got a mowed ditch, you've got the road on the left side, you've got a ditch that's all grass, and then you've got another corn crop. I don't see much there for bees to forage on. On the other hand, just a few miles up the road, I found this field, and I would really like to see a lot more of these. I didn't go through and figure out all the varieties of plants that he has in here, but this is really what we need for pollinators. So I'm not going to spend a whole lot of time, but because this is standard feeding instructions in the spring, feed basically a 1-to-1 sucrose to water. And I think that's pretty standard for just about everywhere that we go. And it's in the same story in the fall, except you use a 2-to-1. And when we talk about a 1-to-1 and a 2-to-1, I've always had the question, I've often had the question, should that be weight to volume or weight to weight, or how do we get to that 1-to-1, 2-to-1? And my comment is always that this is bathtub chemistry. This is not lab chemistry. The bees don't really care whether it's a 1.1 to 1 or it's a 2.1 to 1 or a 1.7 to 1, as long as you have roughly a 1-to-1 in the spring and you roughly try to double that for fall feeding. And then again, after that first frost in your area, then you can change to dry feeding inside the hive. The reason I say that is if you use outside feeders, you know, the Boardman type feeders or community feeders, when it starts getting cold and that syrup gets cold, it takes a lot of calories for those bees to warm that back up when they get it inside their bodies. That's a big calorie push for them. So if you can keep that, again, once it gets cold, go to the dry feeding. Now, I use what we call a Krabby Patty, and I make that up in 5 or in 1 gallon Ziploc bags. And when I do that, I use 4.5 pounds of sucrose, 1/2 pound of protein supplement, and this is for winter feeding, 8 ounces of water, you know, 7, 8 ounce, 9 ounces of water. And I use a tablespoon of Honeybee Healthy. I use a tablespoon of white vinegar, not apple cider vinegar. And I use a teaspoon of salt. And blend this to a peanut butter consistency. I've got a pretty good wife who lets me use her KitchenAid mixer, and that works quite well. Once you get that to kind of peanut butter consistency or a fairly stiff consistency, and that's determined by the amount of water that you put in there, then you can put that into 1 gallon Ziploc bags. And I use small boards, and I've got a picture of this in just a second, to make a rectangle between these two boards that you put in there, and then place those over the frame so that these bees can access that food without breaking cluster. I recommend that the low protein patties during the winter or anytime it's too cold for the bees to make cleansing flights so they can defecate outside the hive. And too much protein during this time when they can't get out and make the cleansing flights will increase susceptibility to nosema because of the overload of nitrogen in the gut. So during the winter feeding, that protein level needs to be about 4 to 5%. And that's where that 4.5 pounds of sugar, the 1/2 pound of protein supplement. And I do that because most of the protein supplements on the market are in that 40 to 45% protein range. Now for spring and summer feeding, the patties should be 18 to 22% protein. And this is where you change from 9 parts sugar, 1 part protein to 6 parts of sugar and 4 parts of protein supplement. So that takes you up to about a 18 to 20% protein in these summer patties. I'm sorry, you know, in the summer patties. And then I also use the Honeybee Healthy, and that is a lemongrass spearmint blend as an appetite enhancer. I think that is its primary function, something to attract the bees. I use white vinegar because acetic acid is a normal component of bee bread, and the fermented pollen is used for winter feeding and early spring feeding. And one thing about the acetic acid is in the bee bread, again, that's somewhat similar to yogurt or sauerkraut or the silages, and acetic acid is the normal component for the normal preservative in here. I use a little bit of salt. There's no clearly demonstrated need in the published literature that I can find, but every species that I know of needs salt. And typically, salt content should be between 0.3 and about 0.5% of the diet, at least for mammals. And I'm assuming it's going to be somewhere in that same range for the honeybees. So this is why I do what I do. Some people have asked me about other essential oils. Again, it goes back to making sure that you've got good record keeping systems and you can evaluate very closely as to whether there's an economic benefit to some of the other essential oils that you might want to try in your bees. And again, we've already talked to probiotics. So those are options that you can add to this. Okay, here's what I use for a Krabby Patty. Here's my gallon. Let's see if I can get my pointer to work here. There we go. Okay, this is the gallon Ziploc bags. These are 3/4 inch by 3/4 inch by maybe 3 to 4 inches. And I put those, kind of equally spaced. And then I cut a rectangle through here, this area right through here. And when I put that on top of the frames, I make sure that the opening that I've got cut here covers at least 2 frame openings. And I try to get that as close to the center of the frames as I can, close to the center of the colony, because when they form that cluster in the winter, they typically will be pretty close to the center of that hive box. And this way they can go up into this Ziploc bag without breaking cluster, or I should say with minimal breaking of cluster, minimal chance of the bees getting too cold, because whatever heat they're producing is rising right into this bag. Come spring, it is easy to clean up. You just throw the whole thing away and you start over next year. I've had colonies go through this 5 pounds in a month. I've had colonies go through maybe half of this throughout the whole winter season. So it will vary, and you have to be checking these hives, or at least my recommendation is checking these hives at least once a month during the winter months. You find a day when it's, well, like the next few days here in Sioux Falls, it's going to be, I think, Friday, Saturday, Sunday, it's going to be in the upper 40s, maybe even to 50 degrees. That would be a good day to go out and check your bees. And then the challenge is right after that, the Monday night, it's going to be 5 below zero. So this is one way for the bees to have extra food, extra insurance, to make sure they've got plenty of food over the winter, and they don't have to exert a lot of extra energy to break the colony and get up into there. That has been very successful as my feeding method for winter feeding. And then over this, I put a quilt box. You can also use patties on here. If you want to make them up as patties, that's fine. Just use wax paper on either side of it. Make sure it's only about 1/4 inch, maybe 1/2 inch thick. That works fairly well too. I like these because, again, they don't have to break the colony, the cluster near as much during the winter. And when I check these, when I have to replace them, I do a real quick check to see if I have to replace them, 2 or 3 seconds. Just lift the cover and see if I have to replace them. And then if I do, then I do the cut in the Ziploc bag, and then I can be in and out of that hive. And even when the temperatures are in the 30s, I can be in and out of that hive in about 10, 15 seconds. And I would also caution you to wear your veil, because I have been stung every month of the year doing this. So it's very effective. But it's also, they're not real happy when you open that colony when it's cold outside. You can use liquid feeding. You can use the frame feeders. That's the main thing there, is put something in there, wire or something for the bees to land on so they don't drown. That's very hard for them to get to that in the wintertime when they're in a cluster. You can use the top feeders. This is a Dadant feeder. Putting the syrup there, they can come up through that center opening. But again, they have to break cluster for that. And if you have a quilt box on top, like I typically will use, maybe it's not so bad, but I think this is fine until you get into probably sub-freezing or even sub-zero temperatures. And then I don't think that's going to be a very good way to feed them. So what happens in the winter? Well, the queen slows down egg laying. We know that sometime after the first frost. In most areas, by mid-November, the queen has usually stopped laying. Where Melanie's at in New Mexico, it maybe later than that, where some of us are in the northern climates, it's going to be, it can be late October when this happens. When the queen stops laying, the brood will let that hive temperature drop down to about 75 to 80 degrees. And that's when they form the cluster. The outside temperature, or they start forming colonies when that outside temperature drops to about 45 to 50 degrees. They do that to stay warm. So the bees will shiver and generate body heat, and they do this by, we'll call it disconnecting their wings. They still flex their flying muscles in the thorax, but there's no wing movement. And this is how they generate the heat. And as a big ball of bees, they just keep moving in and out of that cluster to keep the queen warm. One of my concerns is if you have too small of a colony and that queen gets cold, but the colony survives the winter, I don't know how well a chilled queen will lay. I don't know what that does to the longevity of the eggs that she has stored in her. So that's one of the reasons that I really want to make sure that we keep the queen in good condition all winter long. Bees don't sleep, they do rest, and that's all seasons. If it's over 45 degrees or thereabouts, the bees can make cleansing flights. And I've even seen them make cleansing flights when it's about 30 degrees, but 30, 35 degrees, if the sun is hitting the front of that colony. My question is, if they go out and make cleansing flights, do they make it back? And I would guess that there's going to be a fairly significant number of them at 35, 40 degrees that don't make it back to the colony. Okay, ventilation is essential to maintain the colonies and to minimize moisture buildup. And the moisture comes from the normal breathing and from the metabolism of sugar. That is, whether it's the carbon dioxide in water, whether we're breathing it or the bees, it's the same story. And I use quilt boxes. And if you are not familiar with quilt boxes, it's basically a super that you put a floor across about halfway. You drill some ventilation holes on the side just below your cover, put screens on those from the inside. I use burlap to make that floor. You can use burlap or you can use screen, and then put wood chips on top of that. The moisture and the heat will rise up through there. I've had mold growing in those wood chips in the spring. But the first few years when I did not have quilt boxes, I had water stains on the sides of my colonies on the inside box of the brood boxes, and wet bees being dead bees. So they can stay warm if they can stay dry. So, and you may also see mold growth on the inside of the hive if you have excess moisture. So the quilt boxes, I had much higher winter survival rates when I went to the quilt boxes. I also did insulation wraps, and I have a picture here in just a second that I'll show you right there. Now these are actually on stands. They're about 18 inches off. The bottom is about 18 inches off the ground. I wrap these with, I used an aluminum bubble wrap, and then put black plastic around that. And then I tied that on with twine. And you can't see the quilt boxes very well, but see if I can get the pointer. The quilt boxes are right here. You can just barely see them right below the cover. And I have the ventilation holes over here on the sides, just below the top cover, so you can still get ventilation or the air movement through there. And with this snowstorm, I come out and I wanted to make sure that I had the opening here on the front of the hive, so it wasn't blocked, so I could get air circulation through that hive. This much snow on top makes for pretty good insulation. Now, this is not my hives, but a friend of mine, the traditional Langstroth hive. He goes 3 supers deep, and then he's got a quilt box. No, I don't think he's got a quilt box. I think he's got a 4th super up here. He put this in a shed. This is an open shed, a pole barn, so this still gets the sun for most of the day. And this is the infrared picture of that. This is the degrees Celsius from the cold to the warm to the cold here. That's out here on the scale. But you can see how the bees are up in this top box, pretty well forming a cluster right here. That's why I like to put that Krabby Patty right on top, right through here in the center of that hive. This friend of mine also does, he's gone from Langstroth hives to mostly horizontal hives, and has had good success because he's got at least 1.5 inches of styrofoam insulation all the way around. And this is what it looks like on an infrared picture. You see that now in the horizontal hives, you typically will have a blockade or a frame that blocks off how far over the bees can go, so they only have to heat up one portion. They don't have to heat up the whole hive, but you can tell where the bees are up here. And that's where he said he didn't have to do any supplemental feeding in these hives during the winter. And this was in West Central Illinois. But it's kind of interesting to see where the warm heat sources are. Okay, so the queen will slowly start laying eggs sometime in mid-February, mid-January, February. And again, when she starts is really going to be dependent upon your latitude. You get guys in Montana and the Dakotas and Minnesota, Wisconsin, they're not going to be, it's probably going to be early March, mid-March before they start laying. You get down to where Melanie is in New Mexico, and the queen may never stop laying eggs. But usually when they sense the longer daylight hours, that's when, and they sense warmer temperatures, that's when they start laying eggs. And as soon as she starts laying eggs, that internal hive temperature is kicked up by the bees to about 94 to 95 degrees. And this creates a huge increased demand for stored honey consumption to meet the big increase in energy demand of the energy of the workers. And this is the most critical time for colony survival over the winter. And this is when most colony losses occur. And this is the reason where, especially this time of year from here on out, you really need to be checking your food supplies. If you have a starve out, this is usually when it occurs. And this is what a starve out looks like in my book. All you see is bee butts. And if this is what you see, you as a beekeeper are at fault, because the bees have run out of groceries. And there's something that you as a beekeeper could have done about that. This happened to me my first year of beekeeping, my first winter, and I swore that would never happen again. And so that's why I took pictures that first year, because I never saw this after that, after I started winter feeding. But I did see this on occasion, and this is what I would call a freeze out. And the reason I call it a freeze out is you can see that there's down through here, there's a few bees, all you can see is bee butts. Let me go back up there. But the rest of this, it certainly appears that they were in a cluster and they broke the cluster, had a very rapid drop in temperature, and then it warmed up again, because you can see mold growth down through here, even some mold on some of the bees. To me, that's a freeze out. And I guess the only way that I know to help reduce that is to go to horizontal hives, or much more insulation on your Langstroth hives. The metal bubble wrap insulation that I used on my hives was not adequate to stop this. I had some guys there in Central Illinois in our group that went to the pink styrofoam insulation. I think most of them either used 1 or 1.5 inches and used that to go around their hives, and they did not see this as often as I did. So as you get into the colder climates where you have more winter weather, hive insulation is something that you may want to consider. Okay, now let's talk about plants. And we talked about the 6 families, the rose, the legumes, the figwort, the mint. I don't remember the other 2 right off the top of my head, but most of these important food plants are typically considered weeds, and some are considered noxious or invasive weeds, and they're sprayed for to get rid of. But dandelions are a great pollen source. It's not so much that it's a great pollen source, but it's readily available. It's there all season long. Clover, especially the white clovers, the wild mustards are the mustard families are always good. The deep-throated flowers are not easily utilized by bees, and this includes alfalfa. They're utilized only when other nectar sources or pollen sources are limited. And most grain sources, or most grains, I should say, are wind pollinated, and they are typically visited by bees only when other foraging opportunities are very limited. And again, this is the corn, the oats, barley, wheat, rice, quinoa, and then some of the other ancient grains. And based on eye structure, the bees likely see red as a dull gray color, and they're not readily attracted to red blossoms. So kind of keep this in mind as you think about your pollinator plot. You want the yellows, the whites, the oranges, the purples, but try to minimize the reds if you can in that blend. Visit places like pollinator.org for the flowering plants that are best adapted for use in your area. The main thing here is to have some flowers in blossom throughout the entire growing season. And one of the best papers I've seen on that is an extension paper by Catherine, I don't remember her name right now. She was at Purdue University. She had a really nice 3-page paper on the plants and when they blossom throughout the year. But if you go to your university extension, and you may have to go to several, if your state doesn't have something like this for the beekeepers, you may want to hit the university websites for the surrounding states and see what they have, because I think there's several universities out there that have a spreadsheet that has the flowering plants throughout the entire growing season. Or look for, and this is true whether we're talking about honeybees or whether we're talking about other pollinators such as the butterflies. You want those plants, you want something that they can forage on throughout the entire growing season. So the next part is to make sure that you include the diverse colors, the fragrances, the shapes. And again, they prefer the blues, the purples, the whites, and the yellows from what I can find. They prefer plants in full sun. They prefer large groupings of flowers, that they're much more efficient at foraging a large grouping of flowers rather than single plants. And most angiosperms, that is the flowering plants, are insect pollinated, and those are the ones that you want. And in going through the coursework with the students in the Master Beekeeper course, those that have spent the money on pollinator plots say that they generally get almost the full return of their investment within the first 2 years, kind of depending upon weather and depending upon honey prices and honey production. But if they establish their pollinator plot, and they have good weather, they have foraging for those bees throughout the year, they usually have a considerable increase in honey yield. And they've told me that it's a pretty good return. Most of that financial return is in the first 2 years. And then after that, they use that extra money for additional pollinator plot area. So minimize the hybrid flowering plots, the hybrid flowering plants. Many of these have been bred to have less fragrance, less nectar, less pollen. These are bred for looks, not for the pollinators. Again, provide the clean, fresh water, even if they prefer the dirty water. Use pesticides wisely. Apply pesticides late in the evening if possible when the bees are back in the hives. Join Driftwatch or other similar organizations that will notify you or are supposed to notify you when they'll be spraying in your area. And the reason I bring that up is 4 or 5 years ago with the Zika virus down in Florida, the health department thought that they would be really smart and they sprayed for mosquitoes all over the place, and they sprayed pretty heavily. And it's my understanding that there were several thousand bee colonies lost down there during that time frame, and there was no compensation to the beekeepers when that happened. So make sure that you're participating in something that will let you know when they will be spraying in your area. And again, I've got kind of a warped sense of humor, and I come across this at a food trade show a couple of years ago, and I thought it was very appropriate. Some people may not see my sense of humor, but bacteria, it's the only culture that some people have. So with that, I'm going to stop and we'll go back to Melanie and go for the questions.
Great. Thank you, Dr. Hill. I was just answering one of the questions here in the Q&A that somebody had. And yeah, that was super helpful. And I will share, just because folks, you made a reference several times, and folks, may not realize, but New Mexico, our lowest elevation is 2,900 feet and our highest elevation is over 13,000 feet. So we have pretty interesting weather and topography. So the lower half of our state is actually quite warm, as you were mentioning, where bees tend to run through their food stores pretty quickly. And in fact, down in the southern end of the state, folks are already, getting their first pollen flows right now from mustards and able to feed. But up here in the northern part of the state, like my bees are, really close to the Colorado border, and it's actually really quite cold. So, and I'll start with, a question kind of based on that, because you mentioned in your area, you're able to go in on those kind of 45, 50 degree days and offer feed. When you go into these hives, you're not actually cracking them open. You're just kind of going into the top and putting the food in and out as quick as possible, or what's your approach to that?
Yeah, all that I'm doing is opening the top of that hive and checking how much food that I have put in there on top of the frames. That's all that I'm checking for. And again, the initial check is probably 2 to 3 seconds. And then if I have to add additional food, as long as I have my Krabby Patty packet ready to go, I can get the old one out, get the new one in about 10 seconds. So
Right.
I try to have minimal time that hive is open. And there's been sometimes sometimes I've had to do that in the mid-20s. So I don't want that hive open any longer than I have to.
Right. Yeah, I often share with folks, once you crack open those propolis seals, the bees can't reseal it. You're going to get a lot of drafts. But that's good. And that's also, a good beekeeper is in and out, even during the warm season when you're doing inspections, as you get more skilled, you can be swift about it, because the longer we have them open and we're dinkering around in there, the more that they have to kind of reconstruct their own space as they see fit. So let me ask a few of these other questions. Okay, one of these is actually from the first presentation, but I think it's really interesting here. This is from Doug, asking, are there fungus or fungi that form on substrates such as coffee grounds or wood chips that are attracting the bee? You were talking about them getting that when you're cutting wood and you see them over there. Do you think they're getting some of that? I guess mycelium is maybe what they're alluding to from other research that's been shared. But is there fungus that's forming on there? Is I guess, in a sense, kind of building on this person's question, does that support fermentation of some of their nutrients that they need?
Well, the real answer is I don't know, but my guess answer is that may be something that they would use in the fermentation of the bee bread to make the bee bread. And again, keep in mind that if it's a proper fermentation with the right yeast and bacteria, you get acetic acid. If it is the wrong bacteria or is contaminated with undesirable bacteria, you may get alcohol as your preservative, in which case that's not desirable. But the mycelium, that it may be something that the bees use to help inoculate that bee bread during that fermentation, or they may actually be using the mycelium as a protein source.
Okay, that's great.
That's speculation on my part.
This one came in also from the previous presentation, but since we have a guest all the way from New Zealand. So in New Zealand, this is from Frank, the bees derive a lot of the honey from tree sources. Would there be any differences in the sugar composition from tree sources? And they noted that you also have sasswood. Did they mean sasswood or basswood?
Or basswood.
Probably.
Yeah, and the answer is yes. In West Central Illinois and some of the areas that I live in Sioux Falls area now, some of the earliest pollens that are available are cedars and maples. And that's what they're going to forage on. And how much is the sugar content in that? I would have to go back to, I think some of that information is in some of Eva Crane's books, but I would have to go back and look that up. I don't know that off the top of my head, but I do know that they will forage on certain trees as they bloom during the year. So yes, that's another food source for them.
Yeah, and there's so much we have to learn too about distinct nectars and what their contents are, what their chemical composition is, for sure.
Well, and
and
another thought on that is that honey locust is one of the early pollens, and there's a pretty good market for honey locust honey.
Yeah, we get that along our river woods, our bosque here in New Mexico, and it yields a really tasty nectar too, or honey. Let's see, next question. Oh, well, somebody would also like to know, Skip would like to know if you're willing to share your contact info.
Sure.
Okay, and I'll put that in the chat because I already copied it and I can paste it in. We have another question on protein supplement in Louisiana. It seems to be attracting and attack. They kind of combined attract and attack, attracting small hive beetles. Do you have any suggestions on that?
Yes. Summer feeding, especially where I was at, you had to be really careful on how much pollen sub or patties, protein patties that you put in the hive, and put in only what the bees would clean up in probably 3 to 4 days. So if you're feeding like a typical patty that's a 1/4 inch thick with the wax paper on each side, what I would do is probably cut that into 6 or 8 pieces and put that in, because yeah, it's great food for the bees, and it's also great food for the small hive beetles. And what I was using to help with small hive beetles, one was the beetle traps, and I would put vinegar, or you could put vinegar in there. I use vinegar in it. You could also, some people use vegetable oil in those beetle traps. The other thing that I used was unscented Swiffer pads. I'd cut them in half and put those in right on top of the frames. And those the bees would chase the hive beetles into those Swiffer pads, and they couldn't get out of them. And then when I changed those Swiffer pads, they went right into my smoker.
Right. Yeah, I've seen that done. Those seem to be pretty effective. I've also seen, unfortunately, queens get their legs caught on those on those pads too. But I've only seen that happen once when I was visiting some beekeepers in Hawaii. Um, we've got a few more questions, and I know we're a little over on time, but this is just great, and we're so glad and lucky to have you here with us. To answer these live. So we'll just ask a couple more. Somebody asked here, can we add propolis to syrup? Or I guess the question is, should you add propolis to syrup? I just heard a talk by Dr. Marla Spivak, and she was saying, I know beekeepers are going to ask that question, and I will wait to hear your response before I share what her response is.
I guess it would, I'm not sure that there's going to be a whole lot of value to that, other than if you, I guess I should back up. There may be some value to that, but it's going to depend on what type of feeder that you're using. That is, if you put the propolis in there, is it going to plug up your feeder? If that's the case, then it's not going to be much value. But if you're doing it in a feeder where they can get to that propolis, there may be some advantage. They may be able to use that.
Yeah, her response was that, it's so concentrated that it's not, probably the best thing to give your bees because of the intensity of those resins and all of the plant oils and chemical constituents in that. But what she did mention too is that that they're finding propolis particles in honey and in wax, and so the bees are coming into contact with that and eating some of it to a certain degree, but as to
Yes.
what quantity, and you're right, it depends on the source, and the availability of that too. So another thing that we really, would be really great to have more research on and to learn more about as well. Let's see here. We'll do at least one more question because I know we've got just so much here. Well, two things. Somebody had mentioned about horizontal hive insulation, and do you recommend like 1.5 inches around the inside of the hive for hive insulation on those horizontal hives? Or
that is typically what is used on the horizontal hives that I've seen, and the reason for that is the 1.5 inches is that's the same dimension as your 2x4s that you're using to build it.
Right. Yeah. And I've known here, like in New Mexico, because we have a lot of top bar beekeepers too. It's interesting because we get temperature swings that are so massive. And so you'll have bees really clustering up, and then of course breaking cluster when it gets warm enough. But what I've noticed with the top bar hives that I've had in the past is I always need, again, some type of upper ventilation port, because otherwise they choke out is what I call it. That CO2 level rises as they're respirating in there. And without, just the natural laws of physics in terms of, heat rising, I mean, the heat rises there, but then eventually it just sort of gets stuck and you get the CO2 buildup and they asphyxiate. So I always recommend folks put a couple additional ventilation ports that are screened, and not too massive. I like to use 3/4 inch drill bit because then a wine cork fits in there. But yeah, there's all sorts of little tips and tricks, and maybe in the future we can have a presentation on some horizontal hive design and management. One last question for you. So this is from Michael. In Montana, how might our winter weather this year affect egg laying? We have had days of 55 degrees Fahrenheit in January, and then just 2 days ago, it was 50 degrees Fahrenheit again. So, I mean, I guess that's a shifting weather question, as we're sort of seeing patterns disappear. What do you think that how that's going to affect the egg laying?
Well, I would hope that the queen is smart enough not to start laying eggs yet. And it may depend if Mike is a commercial beekeeper and he has those hives outside where they're getting the sunlight, and those types of temperatures, and when the day is starting to get longer, she may start laying. But if she does, what's the big factor is going to be to make sure that as those worker bees kick up that temperature in the hive, that they don't run out of groceries real quick.
Right. And that's what happens. Yeah. When they get warm enough, they want to start metabolizing and I actually do the reverse, kind of interestingly. You know, you think you want to bring bees down to lower elevation, it's an earlier spring, but I found that March notoriously gets warm, which starts to, you know, cause the thaw up on the mountains, and then that cold air comes racing down to the valleys. So April gets cold again. I call it the Rocky Mountain whistle. So I actually, anything I have in the valley that I brought in the fall because it was, you know, warmer and I could still feed a little, come March, I actually take them back up the mountain so that they don't wake up too early.
So you don't have those wide temperature swings.
Right. And it kind of chills them out again, because never fails. They'll want to, you know, they get duped. They want to start doing stuff early, and then we always get late frosts, you know. So it's tricky. It definitely can, your own location can be tricky, and hopefully, you know, things that we tend to think are easy will change. I almost feel it's like children, you know, once you get used to it, then it changes.
Well, you know, it's almost more of a question of adapting to the weather patterns on your bee management practices than it is actually what your bees are doing.
Mm-hmm.
Melanie, if I might jump in, since it was a
Sure.
Montana question, we always, the whole state gets a January warm up, sometimes surprisingly warm and so on. Then we generally tend to get a follow up in February where we get a cold snap like we're having now. We didn't get quite as much warm weather in January as typical, but we did hit some 50-degree. But you know, year after year after year, Montana bees, I mean, and I came from a dairy operation, I know January always thaws. There's these warm Chinook winds come through, as we call them, and then it goes back into the freezer in February, and February can bounce around. The books say that queens may start laying in February. We've done some studies where we actually looked at colonies every month of the year for several years in the wintertime. Actually, if you do it quick, you can sneak a look at the cluster. Not the best thing, but it didn't kill them off even in pretty cold weather, because we were taking some samples for mites, but it was years ago. But anyway, I have not seen queens laying in February, regardless of what the weather's like. But come along April, all bets are off. And as Dale said, the biggest worry about the warm periods is that the bees will eat. You know, they get flying and so on, and all of a sudden their food can stuff. Even if they're not laying eggs, they just essentially just start moving and chewing through food like mad because they're flying. So you got to watch January or any of these breaks we get, because they can go through an amazing amount of food in a hurry. The work that Ed Southwick did years and years ago said that there's a sweet spot when it's colds and not too cold. The bees actually cluster tighter, and because of the insulation of their own hairs interlacing and so on, they're pretty well insulated block, and that's when they use the least amount of food. So, but the moment that it either goes really cold or it becomes warm, either one of those two extremes triggers quick depletion of resources. So if you're in Montana with our oscillating January and February, you know, and most of us have been here for a long time, know to run out on a warm day and take a quick peek, just like the Dale was talking about.
Right. Thank you so much, Jerry. Well, and okay, I have to ask this one last question. No more questions, people, but this one's pretty cool. So Chinese beekeepers use hydrogen peroxide, 3% to their syrup for nosema treatment. Does that make sense?
To a point. The challenge is that 3% hydrogen peroxide is going to break down to water fairly quickly, especially when you would dilute it down in a syrup. So I doubt that within probably 24 hours that it's going to have any effect at all.
Right, right.
So the answer is, I guess it depends on how quick it's consumed. And then the other question is, because it's a very strong oxidizing agent, what's that doing to the membranes of the bee as well as the membranes of the nosema? So
That's super interesting.
the answer is I really don't know.
Rob Kramer that worked with us in the 2006 to 2010. He's now back at Duke. He left Montana, went back to the East Coast, but he did some early work on the Leucema serorum, and a up to 10% bleach solution was actually one of the more effective ways of controlling it. Now, he wasn't looking at long-term impacts, so whether 10% would have any effect on the colonies, or certainly in short term didn't have any measurable observable effect. But it did do a pretty good job on the Leucema, much better than a lot of the other quotes treatments.
Yeah,
10%, 10% hypochlorite solution, I don't think may not be as hard on the bees as the hydrogen peroxide will, because my concern on the hydrogen peroxide is the oxidation of the lipid membranes.
Yeah, and he wasn't using hydrogen peroxide, so I agree with you.
There's so much we got to learn, and I'm just going to play my last little thank you slide. Thanks to everybody. Thanks for answering questions, Dr. Hill, and thanks for everybody who stuck with us through here till the end. Thank you so much for
Melanie, if you put my contact information out there, use my email address. It's easier to catch me that way.
Yeah, I put it in the chat.
Okay.
I put your email address. So and just one last plug for our illustrious sponsors who help to offset our Zoom costs. Check them out online if you want more information. And please be sure to put us on your calendar for next month when we welcome Ian Stepler from Manitoba and Cayman Reynolds from Tennessee. So we'll have 2 speakers next month talking about what they do best, beekeeping, right? And with that, good night, sleep tight, don't let the varroa mites bite, and thank you to everyone who's joined us. You can follow Western Apicultural Society on formerly known as Facebook, now known as Meta. You can follow us on Instagram, and on YouTube you can see our past recordings and our future ones coming up. So I'll stop share with that. And yeah, thanks so much. You've got a lot of thank yous in the comment section, in the chats, and I think we answered almost all of the questions, or at least touched upon those different answers. But thank you so much, Dr. Hill. This has really been so informative, and I really appreciate you spending your evening with us.